The overall goal of the following experiment is to examine the influence of toxic chemicals on endothelial cell migration using different migration assays. First, the use of a modified Boyden chamber assay to examine the chemokinesis of exposed endothelial cells is demonstrated. In the next experiment, a wound healing assay is combined with cell tracking to provide additional in-depth information about the dynamics of cell migration. In the final assay, investigation of the mitochondrial membrane potential is demonstrated for identifying the underlying mechanisms of impaired endothelial cell migration. Ultimately, these migration assays can be used to evaluate the effects of highly toxic alkylating agents on the mitochondrial membrane potential and migration of endothelial cells.
The methods can help to answer key questions in the field of wound healing disorders induced by highly toxic chemicals. Demonstrating the procedures will be Ramprasad, Conny Wollner and Steffen Müller, all technicians from my lab.
To pre-coat the cell culture flasks, begin by adding a sufficient volume of freshly prepared gelatin to just cover the bottom of a sterile cell culture flask. Place the flask in a cell culture incubator for at least 30 minutes. Then remove the remaining gelatin solution and transfer the embryonic stem cell-derived early endothelial cells, or EEC, in enough media to culture the cells to 80% confluency. When the cells have reached subconfluence, remove the medium, rinse the cells with PBS, and incubate them in 1 ml of Accutase per 25 square centimeter for 2 to 10 minutes. When the cells have detached, split them at a 1:5 ratio in fresh medium.